Well, good evening, ladies and gentlemen, and thanks very much for joining us for this first of our two Pecha Kucha events, which are part of Explorathon 2020. So, first of all, I'll explain a little bit about Explorathon. So, Explorathon is the Scottish version of European Researchers' Night. European Researchers' Night is European Commission funded. It's the largest public engagement with research event in Europe, and we've got a whole range of activities and events happening digitally this year, but across Scotland. And we'd love for you to tell us what you think about them, and we'd love for you to follow us. You can follow us on Twitter or on Facebook at ERN Scot, or by using the hashtag Explorathon 2020. Also go to our website, www.explorathon.co.uk. You can see all the other events that's happening across Scotland, organised by our partner universities across Scotland. And tonight we have a number of people joining us from across Scotland to take part in this Pecha Kucha event. And I'll tell you about Pecha Kucha in a second, but just as a note beforehand to say that we are actually recording the event itself. So Pecha Kucha is a special format for talks. It's short, it's snappy, it's lively, and it's very visual. So the idea is that each speaker has 20 images and 20 seconds to speak about each image. So their talk lasts no more than 6 minutes and 40 seconds. So it really encourages people to think about exactly what it is they want to say in this very visual and fast-paced style of storytelling, or of talk, I should say, rather than storytelling. But starting us off tonight is Madeline Heap. Now, Madeline works for the Centre for Regenerative Medicine at the University of Edinburgh, and her topic tonight, to start us off, is jumping genes, how viruses slip into our DNA. So, Madeline, over to you.
Hello, my name is Madeline Heap, and I'm a researcher at the University of Edinburgh. Today, I want to talk about jumping genes, or how viruses slip into our DNA. This might not mean anything to you right now, but you can think of viruses as the sneaky girlfriend you learn to live with. Now, viruses' girlfriend, they're pretty different, but I will explain how they actually are comparable. I guess most of us experience falling in love, having a relationship, finding a partner. And you will notice after some time that your partner will start bringing more and more stuff into your flat until at some point, you realize, oh, your girlfriend actually moved in. Now, for most kids, that's actually nice, but you will notice that there are some downsides. For example, if your girlfriend does have long hair, and you will notice that there are hair everywhere. Now, you might ask your girlfriend to tie up her hair to do some damage control, and I guess everyone's happy, right? Now, but let's focus and go back to biology. I guess most of you noticed that your hair does look different from your skin, which looks different from your toenails, for example. And this is because each compartment is made out of a different type of cell, a different cell type. And if you look at it under the microscope, each cell will look different if it's in your brain, in your skin. Now the question arises, how do cells know what type of cell to be, right? So similar to humans asking themselves, who am I, and relying on self-help books, a cell might need some book to know who they are, what type of cell they to be. And this is exactly what they do have. They also have some kind of book inside themselves, the book of DNA. If you open up the book, you will see sentences which contain information and tell the cell what to be, what to do. In this context, we call one sentence a gene. And this is a universal concept in the whole, in nature, basically. And not only we do have DNA, but so do plants and animals, bacteria, and also viruses. Now, viruses are quite sneaky, and they actually manage to kind of hijack our DNA book. As I mentioned, viruses also rely on some kind of book to know what they are, which is in this case a virus. But viruses have the ability to actually transfer bits of their book in our book. They have done this super successfully over thousands of years, so that almost 50% of the DNA in each of your cells comes originally from viruses. This is also shown here, where we looked at the DNA that came originally from one particular virus in green, while our DNA, human DNA, is shown in red. And I think it's pretty obvious that huge parts of the human DNA, of the DNA in each of your cells, is originally from viruses. This might sound a bit scary, but in most parts it's actually not dangerous, because our DNA book contains a lot of junk. a lot of parts that don't have any information. So if virus DNA inserts there, we just just ignore it and we don't really care about it. However, virus DNA does have some annoying property, which is they can copy and paste themselves. So they can make a copy which can then, for example, go over an important sentence and destroy some information. And these are called jumping cheats. And this is actually dangerous. Now, of course, cells are not stupid and they develop mechanisms to fight this, to avoid this to happen. So they have something like pins that keep the virus DNA silenced. So we don't have this copy and paste mechanism happening, similar to you telling your girlfriend to tie up her hair. Now, my research is focused on this exactly. Now, in the cell, you don't really have pins. It would be actually quite dangerous. But these pins represent little machines which are resulting in silencing the DNA. These little machines are called proteins, and we have plenty of them in each of our cells. Now, our lab identified one protein, one little machine called SPOCT1, which we have evidence might be important. Now, in order to check if it actually is, we can do a simple experiment where we just remove SPOCT1 from the cell and see what happens. And this is exactly what we did. Now, on the left side, you can see cells with SPOCT1 cells are shown in blue, and on the right side, you see cells without SPOCT1. In this case, we're staining for jumping genes, so this mobile DNA, viral DNA bits. And on the right side, you can see, I think it's pretty obvious that you have a lot of pink, which are jumping genes. Now, in the second experiment, we were looking for DNA damage. It's the same setup. On the left side, we have cells with SPOCT1, and on the right side, we have cells without SPOCT1. And again, on the right side, we do see a lot of pink, which is a lot of DNA damage, which means the cells are basically about to die or already dead, and no one wants to have them in their body. Now, that we know SPOCT1 is actually important, we would like to go further and actually to understand what the function is and what it actually does in the cell. Now, unfortunately, SPOCT1 is a protein, it's super tiny, you can't even see it under a microscope. But what we can do is produce a lot of it and make crystals with it. And this will allow us to visualize our protein. And after some analysis on the right side, you can see we end up with a structure of our protein. So for the first time at this stage, we are finally able to have a look at our protein, our little machine. And looking at this will give us a lot of information. We can look at, for example, the surface, look for holes which might be interacting with another protein, another little machine. And with this information, we can bit by bit put together the puzzle in order to understand the whole system. Now, we know that SPOCT1 is associated with infertility. We also know that the battle against jumping genes is mainly happening in the sperm and egg cells. So we hope by trying to understand this whole system today, we can help more couples to ask themselves the question, how can something so small take up so much space? Thank you.
Well, Madeline, thanks very much for that really good opening Pecha Kucha talk. So, second up tonight is Associate Professor Maria Garaffa, who works for the School of Social Sciences at Heriot-Watt University. So tonight, Maria is going to speak to us about language learning across the lifespan, multilingualism and the benefits of bilingualism. So, Maria, over to you.
Hi everyone, my name is Maria Garaffa and I am Professor of Psychology of Language at Heriot-Watt University, Edinburgh. Today I want to guide you towards this topic. Every language matters for your brain. In my lab, Language Across the Lifespan, we address questions on the effect of language learnings across children, language development, bilingual people, and also old people. The main topic of my conversation with you today is to explore what happens in the brain of a person who has more than one language, a bilingual or a multilingual speaker. Why I'm asking this question? Because if you look at the world population and how many monolingual we have, people who can speak one language, bilingual, trilingual, and multilingual, we will realize that monolingual is actually a minority of our population of speakers. But it's not just that monolingual is a minority. If you look at the amount of languages spoken in our world, you will see that the majority of us speak what is called in this graph others. So languages that are not global, like Chinese, Spanish, and English. This means that they will be bilingual, but bilingual doesn't mean more intelligent. Bilingual doesn't mean that they are smarter than other people. But bilingual or multilingual means something very specific that happens in your brain. In this graph, you have on the left side the connection that are required for language of a monolingual, and on the right side, the brain connections or a bilingual. As you can see, the areas are the same, the number of connections are the same, but the bilingual are just stronger. This is very important because learning a language is not just better for your communication and job opportunities life, but is actually better because it has biological impact on your brain. But we also know that it's so difficult. We know that learning a language as an adult is maybe one of the most complex activity. And it will be so important to look at when do I have this effect on my brain? Do I have to be bilingual? Or maybe, like in the Matrix learning style, I can just be there, passively exposed to a language and get the benefit of my brain without being a proper bilingual speaker. So just listening. Or even during sleep, you know that a lot of learning we already know happen when we just sit and relax. And this is what consolidate our memories. What about languages? What happens in our brain, even if I'm not a speaker, but I'm just immersed in a multilingual environment? I will present some data on a study, recent study, on American people living in California, that is a very multilingual area of the United States, and American people living in Pennsylvania, that is quite a monolingual part of the country. So these are both monolinguals. But as you can see, if you explore the brain of monolinguals, so people who can just speak American English, the fact that they live in a multilingual environment, just this make their brain quite different. So we need to please people that can speak other languages, any languages. If you're like here in Scotland, we have a minority language, Scottish Gaelic. Even Scottish Gaelic can have the effect of the brain and reinforce the brain connections. And this is what we did in a specific study with young adults learning Gaelic at school. We measured their connectivity and we found that even if they don't speak the language outside the school, but just the language is spoken in the school, you can have the benefits of being bilingual. These are very important pioneering studies confirming that any language matters for your brain. Another dialect that actually is a proper language that we have investigated is Sardinian, spoken in an island in the middle of the Mediterranean Sea. We were there, look at people with very low level of education, but active bilinguals, very fluent in both Italian and their local languages. We explored the area in green that apparently in the middle of the island is the one where you have a lot of speakers learning, speaking both Italian and Sardinian. We look at their brain and again, we found the advantages of being bilingual, even more memory improvement, not just on language, and especially in the one who are switching between Italian and Sardinian the most. So the one who are active bilinguals can have benefits on linguistics, but also beyond language, like on memory. This gives you the idea of how important is bilingualism, multilingualism for our brain. With some new research, actually the idea that bilingualism is very good to prevent some degeneration of your brain, like in dementia and more specifically in Alzheimer dementia. So keep calm, try to learn a language, and if you're not good at learning language, just try to stay in an environment where we will be exposed to many languages. This could be enough for you to have the benefit of bilingualism. Thanks a lot.
So Maria, thanks very much. Really good, really interesting talk there. So next up we have Anne-Kristin Kauna. So Anne-Kristin is a Marie Sklodowska-Curie Actions Fellow based at the University of Aberdeen, and her topic tonight is fungal masquerade, how human pathogenic fungi hide from our immune defences. So over to you.
Hi, my name is Anne-Kristin, and I'm a microbiologist at the University of Aberdeen. Here you can see me with my research object, yeast that can infect us humans. You might already have been in contact with a friendly kind of yeast, the one that we use for baking in bread and cake. And the story that I'm going to tell you this evening is also related to food. It's about a hungry opportunist. You probably already guessed that I'm quite fond of food, and I'm not alone, not even when I'm alone at home during lockdown. In my gut, there are millions of hungry tiny microbes that happily eat with me. And one of these hungry microbes is Candida, the yeast that I'm studying. Candida is a distant relative of the yeast that we use for baking, with the difference that it lives in the guts of about half of all humans. In this picture, you can see my group, the Van Hommig group, that investigates Candida in laboratories all over Europe. And I indicated the proportion of people who could have Candida in their gut. None of us actually checked whether we have it, though. In a healthy gut, we have lots of different microbes that help us digest our food. And Candida may be present, but if so, only at very low numbers. And this is how we want it. But before I explain why we don't want lots of Candida in our gut, I'll show you how Candida is usually kept at low numbers. So in our gut, there's lots of food, but there are even more microbes. And this means that competition for the food is really high. For Candida, this means that it's not so easy to be the first at the queue for food, or in this case, burgers. And some of the gut bacteria even use quite mean tricks to keep it away from the food. And then the gut is not what you would call an easy environment. Even though it's not really a desert and there's lots of food, it can be very hostile. So think of the stomach that is very acidic, and just a few turns of gut further down, the pH can be the opposite and be quite basic all of a sudden. And Candida has to deal with that. And then there's also quite a lot of motion as the food that we have processed is pushed out. Candida has to do its best to stay in the gut, and the places to hold onto are often occupied already by the other microbes. So all these mechanisms make sure that the numbers of Candida are kept low, while the numbers of the other bacteria are quite high. And if you'd like to hear more about the friendly bacteria that we have so many of in our gut and how they help us in numerous ways, I recommend you to listen to Nathaniel Cole's Pecha Kucha talk tomorrow. But why don't we want a lot of Candida around? So although Candida behaves friendly most of the time, it can make vulnerable people very sick. And this is why we call it an opportunistic pathogen. It causes life-threatening infections in about 40,000 people every year. It's constantly on the hunt for more food, on this example, water, and if given the opportunity, it will take it. So what are these opportunities that Candida is looking for? Here's the good news. Candida can't affect healthy people. But if a person has had severe antibiotic treatment and lost the protective microbes, or they don't have a functional immune system, for example, due to an organ transplantation or chemotherapy, Candida might use this as a chance to take over. And then it looks like this in our gut. Candida then can grow much better and multiplies, and the friendly microbes are then outnumbered. Also, once Candida becomes evil, it can change its shape and grow in filaments too, which provide it with a few additional superpowers. And these additional superpowers can help it to infect us humans. So for example, it now has more sticky proteins on these filaments, which help it to stop being flushed out from the gut. Being able to stay in the gut now means that it can also eat more, and for that it also has additional superpowers. now produces a set of special proteins that help it collect more food. For example, these are enzymes that break down other proteins or fats and carbohydrates, or even clever proteins that collect special nutrients like metals like iron, for example, from the gut and then bring it back to Candida. And at that point, Candida gets really greedy. So it's growing so much now that it needs more food than is easily available. And so it attacks the human cells too. And for that, it uses a special toxin that makes holes in the host cells so that it can eat what's inside. Pretty gross, right? So the human cells unfortunately die in that process, and this opens the door for Candida to leave the gut and enter the bloodstream and infect other organs like the kidneys, the liver, and the heart too. What a horror story, right? And at this point, the infection, of course, is really bad. So our immune cells, they try to do their best and fight Candida. And what's really cool is that they can recognize Candida actually by one of its weapons, so the toxin that it produces. But still, even though the immune system tries its best, they need help because the person doesn't have enough of them if they are sick already, and Candida can overwhelm them. Luckily, our doctors nowadays have something at their hands that's called antifungals. These are substances that can stop Candida from growing or even kill it. Antifungals work in a similar way to how antibiotic works against bacterial infections. However, as you probably have heard on the news already, antibiotic resistance has already become a huge problem in the past years. Infections that have been for decades now become difficult to treat again, and the same happened with antifungals and Candida. Doctors worldwide observe antifungal resistance increasingly often and are struggling to treat their patients. And this is why scientists like my colleagues and me from the Van group and labs worldwide are studying Candida and how the infection process works. We are looking for new methods how to help patients fight a Candida infection or how to make sure that Candida just stays friendly in our gut. So now I want to thank you for joining the Pecha, Kucha night this evening in the socially distanced manner and to listen to my presentation. I hope you enjoyed the story and I hope it has provided you with some food for thought. Stay safe.
Okay, thanks very much. Again, really interesting talk and really sort of highlighting the role that fungi have to play within the human immune system there. So next up we have Dr. Tom McKean. So Tom works for the Elphinstone Institute at the University of Aberdeen, and his topic tonight is the international influence of the ballad and song traditions of North East Scotland. So Tom, over to you.
My cunning plan is to submit you to a war of attrition now, to bombard you with some evidence that this is the ballad capital of the Wardle, and the place where you can find the best ballads and the best songs in all the world, right here in this little pocket of the Northeast. This is a map of the Greg Duncan collection, places where Greg and Duncan found songs. This is a family of the Stuarts, the Traveller family, and I just want to talk a little bit about songs and how they work in families. They're passed down within families, and they join families together. Sometimes they separate families, but they tell the history of a place, the social attitudes of a place, how people feel and think about things. 200 years ago, people started looking around all of Europe looking for songs because that was how they would identify who they are, where they belong. People were tired of sort of pan-European, pardon the idea, pan-European identity. So they started looking for their own roots that weren't Greek and Roman roots. And one of the prominent collectors around up here was Peter Buchan from Peterhead. He was trained himself to be a printer and went around the countryside collecting songs from people. He published about 300 ballads, wonderful ballads, and where he was missing for a verse or two, he added a verse or two. As Gavin Gregg said about him, he was never fashed. Then this American scholar, Francis James Child, joined the club and collected 305 different kinds of ballads, many of them here in the Northeast. Most of his texts, 100 of his 305 texts, his favourite ones are from the Northeast here, 37 of them from Buchan. This is Gavin Gregg and James Bruce Duncan, 1904 to 1914, set out to scratch the remains of song, maybe to get 20 or 30 songs from the countryside. They ended up 10 years later with 3,500 songs. So the tradition wasn't exactly dead. And that is now published in 8 volumes. This is an American collector, 1930, James Madison Carpenter. Didn't know the weather here, he bought an open-topped car and traveled up from England, and it got colder and colder, and I just went into a shop, he said, and I asked for the thickest, longest woolen long underwear you have, and I wore it day and night. And he slept in his car sometimes as well. And that was the beginning of real recording collecting, audio recording. Alan Lomax came over from America, blacklisted during the communist witch hunts of the 1950s. Alan Lomax on the left there. Hamish Henderson, the great Scottish folklorist, came up here and they collected hundreds of songs and ballads, more than 2,000 entries in the School of Scottish Studies. But before that, way back, in Aberdeen, there used to live a little woman named Anna Gordon from up D-side, and she provided some wonderful ballads to Sir Walter Scott and got very mad at him when he published them without her permission. But she was a very interesting crossover. She had these wonderful oral ballads, like this one. Oh, there were 7 brothers, oh, I saw, and oh, but they were wondrous bonny, oh, that but this very night we all mun be hanged for the stealing o' the Earl's a lady, oh. So these ballads tell very, very tragic and intense stories, but they don't tell you how to think about it. Not like Victorian songs which tell you that you should weep a lot right now and you should feel very sad. The ballads just let you sit, they let you experience the stories and ideas that are out there, and you have to make up your own mind, and meaning is created between the singer and you. This is Belle Duncan, one of James Madison Carpenter's contributors, and he said, "Do you know this song?" She said, "Aye." "This song?" "Aye." "This song?" "Aye." And I thought to myself, this woman is certainly crazy. But he spent the whole summer with her, with his typewriter on his knee, 3 months with this woman, Belle Duncan, collecting songs, 65 of those big ballads, and another 100 songs.
3 gypsies came to our hall door, and oh, but they sang bonny, oh. They sang so sweet and too complete, that the stole they hurried. And twa bonny bairnies she has born, down by the greenwood side, oh. And made it long and narrow, and laid it down to fall asleep, and she died for her true lover.
3 wonderful singers who are no longer with us, but now, of course, we have plenty of living singers, lots of young people taking up traditional song and feeling that deep connection with place, that deep connection with human emotion, that deep connection with family connections that are found throughout folk song when they're passed on from one person to another or experienced in groups like this. And of course, they didn't just stay here. The songs are far too good to stay here. They went over to America with settlers of various kinds over hundreds of years, down the Appalachians and up in the Canadian Maritimes, and found their own way there, became country and western, mixed in with blues, became rock and roll. And then what happens after that? They all come back as different kinds of songs, country and western songs, skiffle music, which gave rise to the folk revival here in Scotland. And then, so the songs are recycled into new forms, then that breeds a revival in the older forms as well. People get interested all of a sudden in what's right here, rooted in their own place.
Well, it's what's keeping my love so long. Oh, hangman, hangman, slacken up your line. For all this rain is silver, even when the God is so long. Hangman, hangman,
Led Zeppelin there at the bottom. So here are some people who have some Northeast connections with their music. Neil Young, family from Fogie. Joan Baez's grandfather was a minister at Rothy Norman Kirk. And Johnny Mercer there, the composer, with Pitsliga connections. There's a lot of musical connection here. It's gone all around the world, and now it's home, and we should make the most of it and celebrate this ballad capital of the world. Thank you.
Okay, Tom, Thanks very much. So we hand over to our final speaker for tonight. It does, the whole night goes at a fantastic rate. So our final speaker is a research postgraduate. That's Lucya Blaha. So Lucya works for the business school, or I should say researches in the business school at the University of Aberdeen. So her topic tonight is making sense of intelligent automation and digital transformation in the public sector. So Lucya, if you want to take it away, over to you.
Hi, my name's Lucya Blaha, and today we'll talk about AI in the public sector. Now, how many of you would have had to solve a problem using a public sector institution in the past couple of years? Probably a few. Now, did you have to do something like going into a building that looked like this? Like this? Did you maybe have to have a customer appointment? Or did you try and solve everything by using your mobile phone, speaking to a chatbot, or simply having a quick search on your computer? Probably something along the lines of these three. Now, the more we look at public bodies, the more we can see that their interfaces are increasingly digital. They are using more automated processes to fulfill their tasks, and their services are more and more intelligent, at least hopefully. So why have they been doing this? Well, there can be a few incentives behind this. One can be government initiative. Another one can be the need for service continuity due to constrained budgets. But the biggest one is our demand, our expectations for services to be increasingly digital and faster. Should we be worried about this? After all, we know technology has misjudged people's emotions, thought some races are more likely to be criminal than others, and even mislabeled people in a very denigrating way. We could say that technology has oftentimes led to harm. What do these people have in common? Well, they have in common the fact that they've been harmed in some way by the misuse of technology and the misprogramming of technology that could have otherwise been used for good. A lot of organizations are, however, trying to get it right. A lot of public sector bodies are taking a lot of actions to try and improve initiatives, programs, and to try and put these technologies under scrutiny. Additionally, we have celebrities endorsing this and actions from the economic forums. There are talks of ethical concerns, business practices, and design considerations to adjust these technologies and use them for the public good. So why should we care? Well, private enterprise can show us plenty of examples where things are going wrong at the moment. Technologies that could be quite useful have been used for theft, have contributed to people being arrested, and have led to biased consequences for innocent people. So we can see more and more initiatives of governments looking at things they can do to improve the use of AI and to regulate how this is implemented in public services. Well, again, why is this relevant to us? It contributes to a different relationship. By interacting with public services, we interact with organizations that are obliged to respond to our feedback, that take into account considerations of harm, initiative, and consequences for the public community. By engaging them, we can turn things from this to this and increase the amount of collaboration we have and the impact we have on society overall. A response to public sector organizations depends on all of us working together, and the way AI affects our lives greatly depends on it. Why do any of that? Well, the idea is that technology shapes us just as much as we shape it. We spend a lot of the time involved in a system that goes both ways. Us designing technology and us being designed by technology with our actions. We are more connected than ever before, and we use technology to be alone, to be together, and to depend on for critical things such as our health and the expectancy of our life. Overall, we could say that we are more connected in terms of our interests, in terms of our awareness of what surrounds us, and in terms of our direct input. We are no longer disconnected from public sector organizations, from technology, and from others overall. We have plenty of examples now when technology is also being used for good, very many times backed by community responsible organizations to protect us and our health, to use intelligence for good, to manage our infrastructure, and to help our habits improve. So I invite you to play. I invite you to be a part of that environment and to look at the message, platforms you are using, and world around you. Actively engage and find the communities that are relevant to your interests. I invite you to look at things beyond the media content that you might have online, to find what inspires you and to create technology or to contribute to the creation of technology that represents you. And this can be done by engaging with the organizations, voluntary and official, all around. Ultimately, we are part of the world. We do not simply obtain knowledge by standing outside of the world. Thank you very much.
Luciana, thanks very much for that. That was a fantastic finish to our Pecha Kucha event for tonight. So we hope that you've all really enjoyed these talks, and this is the great thing about Pecha Kucha, is you hear from the best and the brightest about their research, about their own personal journeys, and you hear about so many different and diverse areas. So tonight's been a fantastic night. I'd like to draw your attention to the QR code that's on the screen, and if you follow that, it will take you to a feedback survey, and we really encourage you to do that if you have a moment. It really helps us improve as we go along. So in the meantime, I'd like to thank all of our speakers for their time and for sharing their fantastic research with us. I'd like to thank everyone, ladies and gentlemen, who came along and joined us tonight. I hope you've enjoyed it. We do have another Pecha Kucha event. Do follow us on Facebook and Twitter. Look at our web page as well, www.explorathon.co.uk. We've got a whole range of other events, so hopefully we'll see you at some of them, if not the next Pecha Kucha event, which is tomorrow night. Otherwise, thanks very much and bye-bye.